So far, in our survey of molecular transport, we have focused almost exclusively on the movement of some sort of substance across the plasma membrane. So in and out of the cell and how it gets across the plasma membrane to move in and out of that cell. We're going to expand our viewpoint a little bit and take a look at the movement of substances across a layer of cells. Now, notice that if I'm going to move something across a layer of cells, I actually have to move it across two membranes, like so. Any type of transport that occurs across a layer of cells, and that layer of cells is epithelial tissue, is referred to as transepithelial tissue. So transepithelial means across epithelial tissue, and it's a type of, transport. Typically, transepithelial transport involves moving from an external environment to the extracellular fluid. Now, when I say external environment, it could also mean something that's still technically inside of what you would consider to be your body, like inside your mouth, inside your nasal cavity, inside your digestive system, your digestive tube, even inside your lungs. That's all external environment. And so if you eat food, for instance, you're going to need to move those food molecules, let's say my X is a glucose, across a layer of cells lining your intestinal tube into the body in order to capture it. That's absorption. Likewise, if you are breathing in air and you're trying to capture oxygen, you're going to need to move it from the lung airspace into the body. And so that's transepithelial transport. You'll notice that I have 3 different morphologies of my cells. They vary in their height. So on the left side, I really have a nice short cell. In the middle, I have kind of a cube-shaped cell, and then my elongated cells are going to be on the right. If you've had anatomy, you know these as simple squamous, simple cuboidal, and simple columnar, respectively. Those are not terms that I use, but I did want you to make that link if you've had anatomy. So here, we really look at the structure-function relationship. Each type of epithelial tissue is designed for the type of transport that it needs to do. So let's consider the far left example. Simple squamous epithelial tissue, where the cells are thin and kind of flat, is best used when you're trying to maximize diffusion rates. So what do I mean by that? Well, let's consider the following example. Let's assume that I am going to line the inside of my lungs with some sort of epithelial tissue. And I'm going to expose my lung airspace to a certain amount of oxygen, and I want to maximize the amount of oxygen that makes it into my body, so across both layers of cells, as quickly as possible. Let's talk about diffusion rate. We've learned about some things that affect diffusion rate. They are temperature, molecular size, distance, and concentration gradient. Temperature should be around 37 degrees. That's not really going to change too much. Oxygen gas has a set molecular weight. That's not going to change. And we can change the concentration gradient by increasing our breathing rate or our ventilation rate. That'll be discussed more in the pulmonary lectures. But here I'm looking at distance. Which one of these has the shortest distance between the external environment and my interstitial fluid? And it's this one, the simple squamous cells. In other words, I'm minimizing the distance, and therefore these guys are going to move over faster because they don't have as far to go. Compared to these, which have a farther distance to travel, and especially these over here. These have the farthest distance to travel. So if my goal is to maximize the diffusion rate across epithelial transport, then I really want thin, flat cells. I want simple squamous. So simple squamous epithelial tissue is designed to maximize diffusion rates. And this works best for things that diffuse through the phospholipid bilayer or exhibit simple diffusion. And oxygen gas and carbon dioxide gas are two of those substances. So it would make sense then that where we want to maximize the diffusion rate of oxygen or CO2, that we're going to use this simple squamous epithelial tissue. And indeed, in anatomy, you are taught that the lungs do in fact use simple squamous epithelial tissue, the one that is here on the left. Okay, we tend to see the simple cuboidal in the kidneys, and then we see this one in the digestive system. So what we're going to do now is kind of take a look at the far right idea, where transport is highly regulated and controlled, and we're really not using simple diffusion. We're going to use facilitated diffusion and or active transport in order to move molecules from the external environment into the body. So let's first draw something out. If you're going to draw this with me, give yourself a full page, probably turned landscape style, and draw out 3 kind of elongated cells. I have here some undulations in the top of my cells. You don't need to put those undulations if you don't want to. I'm drawing them in because I'm imagining that this is the digestive system, and the digestive system indeed has these undulations in the top part of their cell. Those undulations are referred to as microvilli. So pause the video and go ahead and draw this out. All right, the first thing that we need to do is we need to make sure that whatever we're trying to transport across our layer of cells does not go through the cracks in between the cells. So I need to make sure that I have the appropriate intracellular junctions that create a tight seal, and those are going to be tight junctions. So let's go ahead and add in some tight junctions. Tight junctions are always used in epithelial tissue to seal the plasma membranes of 2 adjacent cells together. Okay, the other thing that we need to understand is that there's 2 membranes that we have to cross. If I want to transport whatever this purple molecule is to the other side, I am going to need to cross 2 membranes, one here and one down here. So we need to give those membranes names. The membrane that faces the external environment is always referred to as the apical membrane. The word apical has its root in apex, which means top. This membrane that's down here, the second membrane that substance will need to cross, is on the bottom. And so this one is referred to as the basal membrane if we are actually down here on the very bottom. But notice that it kind of comes up on the sides. So to be on the side of something is to be lateral. So sometimes those concepts are joined together, and we call this membrane the basolateral membrane. So that's going to be the kind of the bottom-ish membrane. Okay, I want you to envision for a moment that we are going to move this purple molecule across. As I've drawn it, it is concentrated in the external environment. So let's actually go ahead and add in our compartments. So remember in a previous lecture when I talked about the cell wanting to be very economical? That is to say that the cell does not want to pay for transport that it can get for free. And so if you are moving from an area of a higher concentration to a lower concentration, that should be free transport. That should be passive. But if you are moving from a lower concentrated area to a higher concentrated area, that's going to be active transport. In my drawing here, I have a pretty strong concentration gradient of purple dots because they're concentrated in the external environment, whereas there's none in this intracellular fluid and there's none in the ISF. So if I want to cross the apical membrane, is that going to be active or passive transport? Well, that's going to be passive transport because I'm moving from an area of higher concentration to lower concentration. Okay, now what I've done is I have modified this picture, and I'm looking at sodium and I'm looking at glucose. So my red dots are going to be sodium and my blue hexagons are glucose. Let's first evaluate where the concentrations are. At first, I'm only going to look at sodium. So from here to here across the apical membrane, do you think that's active or passive? That's going to be passive because I'm going from a higher concentrated area to a lower concentrated area. Notice that there's almost no sodium inside the cell, but there's quite a bit of it in the external environment. So I should not have to pay for that sodium transport. But whatever I move in the cell, I'm also going to have to move across that basolateral membrane. So now, if I move a sodium into the cell, let's just go ahead and pretend I've moved one into the cell. I'm going to have to move it across that basolateral membrane as well. Now let's take a look at this one. Is that active or passive transport? That's going to be active transport because I'm going against a concentration gradient. I'm going from an area that has low concentration of sodium to high concentration. So I'm probably going to have to pay for that. The way that I almost always pay to take sodium out of the cell is with the sodium-potassium pump. So I said we would see that membrane transporter over and over and over again. So let's go ahead and add that in. It's kind of a ubiquitous transporter. This is our sodium-potassium pump. I'll put one in every single cell here because that's what kicks the sodium out and keeps the sodium levels low in the cell in the first place. Now technically, I should be dealing with potassium, but I'm ignoring potassium in my current image. So yes, potassium is pumped into the cell, but I'm not really considering that at the moment. So the sodium-potassium pump will continue to pump sodium out of the cell, keeping the sodium levels in the cell quite low. Okay, well, so what could I do up at the apical membrane to let sodium into the cell? I could put a sodium channel. That would work because sodium would naturally be compelled to come into the cell. But before I go ahead and do that, let's take a look at glucose first. Glucose is most concentrated inside the cell. So if I try to move glucose from the external environment into the cell, is that active or passive? That is active because I am moving glucose against its concentration gradient. So I'm going to have to pay for that. Now what about getting glucose out of the cell into the interstitial fluid? Now I'm going from a high concentration of glucose to a low concentration out here. So that's passive. I should not have to pay for that. So let's first look at moving glucose across that basolateral membrane. Glucose cannot diffuse through a phospholipid bilayer. It needs some sort of facilitation to diffuse. Am I going to use a channel protein or a carrier protein? I'm going to use a carrier protein because glucose is too big to use a channel protein. So I have to use a carrier protein. Is that carrier protein going to have to spend ATP in order to move glucose from the inside of the cell to the extracellular fluid? No. Glucose naturally wants to move out of the cell. All I need to do is give it a carrier protein that will allow that movement to happen. So the carrier protein that I'm going to put in is called a GLUT carrier. It does not use ATP. It is passive. So that's going to allow glucose out of the cell. All right, we've looked at transport of both sodium and glucose out of the cell across the basolateral membrane. Now, let's return to the apical membrane. Moving sodium into the cell should be passive, but moving glucose is active. I guess I could pay for the movement of glucose with ATP, but that might not be the most energetically efficient way to move glucose into the cell. In a prior lecture, we learned about the sodium glucose cotransporter, where sodium is allowed to diffuse down its concentration gradient and it brings glucose with it. You think maybe I could use something like that here? And the answer is yes. So instead of just putting in a sodium channel and a glucose transporter or a glucose pump that I pay with ATP, I'm going to move both sodium and glucose together using a sodium glucose cotransporter, which is a type of secondary active transport. So let me go ahead and put that one in here. I'm going to move both of these into the cell together. And I don't have to expend any more ATP. So hopefully this image here shows you that the transporters that you insert on the apical membrane are usually different than the transporters that you insert on the basolateral membrane. What I just diagrammed is shown in a picture in your book here. So you can go ahead and pause and go ahead and take a look at all of this and make sure that it matches everything I just said. Okay, on my exam, I will expect you to be able to predict if movement across a membrane is active or passive based on the concentration gradient. Remember that if something is going down its concentration gradient, so like from high to low, that's passive, but if we're going from low to high, that's active. And I want to be able to pay for all transport with as little energy as possible. So utilizing a situation like this where I'm coupling the diffusion of one thing with the active transport of another is a cheaper way to move both sodium and glucose over paying for glucose separately and just letting sodium diffuse through a channel protein. Also, note that the transporters that are on the basolateral membrane typically are different than the transporters in the apical membrane. In addition, you should be able to identify the apical membrane, that these are tight junctions, and the basolateral membrane, as well as external environment inside of the cell and extracellular fluid. All right, that concludes our look at transepithelial transport. In our final lecture, we are going to be looking at osmolarity and tonicity.